So I'm Jonah Metalcats, this is Ian Livingston, and this is Sartaj Ball. And our presentation is about the connection between the gut microbiome and the immune system. So a little background on this is that a microbiome is a community of microbes, which are microorganisms found in the body and nature. And we are focusing on the human gut microbiome. And this kind of focuses on the grand challenges we've been learning about this week, which is to study the microbiome in humans and figure out how the microbes there interact and how we can use this to impact human health.
All right, so our question is how do gut microbes impact the immune system? Scientists have found a link between microbes in our gut to the immune system. The importance of this is to improve vaccine efficiency and discover the cause of ineffective immune systems in our population. Our proposal is to further studies on the microbes in the human gut in order to see the full effect they have on the immune system. Our plan, Our plan is to approach this issue by comparing microbes of the healthy immune system to microbes in the unhealthy immune system by using confocal microscopy.
So for our method to approach this topic, we thought confocal microscopy would be the best. So just a general description, confocal microscopy, it's shown in this image here. It has a pinhole right next to the lens, which blocks out the light above and below the sample. So it allows you to make three-dimensional cross sections, so giving you a more physical rendering, so it lets you look inside other samples that you have. It's particularly useful for 3D rendering and observation of samples below their surface, which is often what you need to find out exactly what is causing the deficiencies or strengths in immune systems. And links have been found between beneficial microbes and the production of T cells. And hopefully soon they'll also find links between B cells as well. T cells are cells in the immune system that directly kill disease cells, while B cells produce antibodies which counteract diseases that have been introduced into the body already.
Okay, and then moving on to social context, which is basically how this can be applied to the general population outside of the lab. So we already know that gut microbes affect vaccine efficiency, and further understanding these microbes can help us improve these vaccines to a higher level. And then targeting the immune system through these microbes can enhance research and prevention for diseases like HIV and AIDS, which weaken the immune system. And we know that gut microbes are influenced largely by your diet and what you eat. So hopefully studying the microbes in the gut can help create diets for the public that can strengthen their immune systems.
So there's been multiple studies in recent years exploring the connection between gut microbes and the immune system. So in 2014, the University of British Columbia began researching asthma, allergies, IBS, and diabetes. So they found initial signs that the composition of microbes can lead to failure to fight these diseases. The Weiss Institute of Harvard built upon this and focused specifically on IBS. So they have presumed links that microbes, epithelial cells, and the immune system are related, but as of now, they have no definite conclusions on why. So that's a field that can be researched further in the future.
So our project proposal is to continue using confocal microscopy to study the human gut microbiome. Using our data, we want to continue making links between the gut microbiome and the immune system and build off of preliminary studies on non-human subjects to reach a cure. Our main goal is to fully comprehend this connection in humans and use it to our benefit.
So as of now, there are multiple challenges in approaching this problem. So while it's more straightforward to look at similar microbes in healthy or particularly weak immune systems, it's very difficult to figure out exactly what gene causes the strength or the weakness. So once the particular microbes have been isolated, you have to find what genes are present using confocal microscopy, which presents a challenge. An Imperial College paper outlines the link between microbes, diabetes, and obesity within mice, but they have yet to bring this particular instance into the human scale. So most early testing begins with animal test subjects, but a challenge is that it's difficult to convert from the animal test subjects to humans as well. Yeah.
Okay, so to wrap this up with the discussion, our current proposal focuses only on the associations between the gut microbes and the immune system and finding remedies for the negative effects caused by unhealthy microbes.
Our future direction consists of increasing our scope of research about gut microbes to include all the major body systems of the human body, so cures can be found for any microbe-related diseases. These goals can be achieved by using confocal microscopes to image the full three-dimensional structure of the microbes.
And then, so we have our contributions, which slides we talked on and worked on, and then the references for our presentation. And we can open it up for questions now if anyone has them.
chain in the way animals digest and utilize their food sources and what they eat seems completely different than what humans. Why would it not go straight to human testing? Because it seemed like it would really affect.
Well, for the example about the mice and obesity is just, I think, to show that a lot of times there's links found between the microbes in animals and their immune systems that we can try and connect in humans. But for human testing, it's mostly done. So if someone's getting surgery on their stomach, you can take a sample from there and use it. But also it's pretty easy to test mice, and because that they found that link, we can hope to find links like that in the future on human test subjects.
So when you guys look at the microbiome, do you expect to find patches of individual microbes, or what would you expect to see as the structure of the microbial community inside of something like the gut?
Yeah, so usually the microbes are specific to each of the different regions of the body, and so we're focusing mostly on the stomach and the digestive tract, so the gut. And we, in general, because it's so specific to the area, that in the digestive tract it will be more spread throughout the body. You can test for what areas they are in mice particularly, just because of the question before, the prevalence. But we don't, assume to find one exact small grouping of bacteria here and over here. But research is still being done to figure out exactly what amounts of bacteria and what types of bacteria are throughout people, and also depends on the person. Everyone has different microbes, so that changes a lot of different things about you. So it's really variable.
I have a question. So when you were doing research on the links between the microbiome and the immune system, did you find anything about different microbes affecting different systems, and do microbes only affect kind of one system at a time, or are there microbes that a single microbe that might affect multiple different systems in the body?
Okay. Well, we found that in humans, especially we focused on the gut microbe because it affects so much of your other parts of your body. Is that what your question is?
So I guess my question is so there might be links between X bacteria and the immune system. would that bacteria also affect other systems in the body? Like if it affects your immune system negatively, might it also affect something else, maybe even positively?
I mean, yeah, that's possible because, like, especially in the like gut digestive tract that we were focusing on, it affects so many other things. Like, it can affect, like digestive, like mood, all these other things other than your immune system. So yeah, that's possible.
Other questions?
I'll ask a technical question. So if you image the three-dimensional structure of a microbe, right, like what do you hope to associate with the actual immune response or deficiency of a person from that knowledge gained, right? So you're going to image the microbe, you see what the structure is. What's the association or the linkage that you want to look more deeply into with respect to the actual immune function of the patient from where those microbes came from?
Well, you could like look at the structure of microbes, like we said in the presentation. You can compare the structure of those microbes to microbes with a person who has a healthy immune system. So within that, you could look at the differences between them and figure out like, oh, so this part of the structure is odd, it's shaped a little different.
Are there indications from the literature that structural differences in the microbes have been associated with these changes, like the examples that you gave of the previous work? Were they looking at the structural elements of the microbes themselves, or were they looking at other aspects?
Yeah, so there's a study done looking at diets especially. So it was comparing the zinc intake of mice, which was linked to CD8 cells within the immune system, it's a lot like B cells. It produces antibodies. So often it's not the exact, physical layout of the bacteria. It's which are most prevalent in comparable test subjects, and also the diet, which is an extremely large part of what the digestive tract entails.
Students? You guys can't ask each other questions. It's a good thing.
Yes.
After those like in your discussion about future areas to investigate, what are some of the other areas of the body that we would like to go to after you've done imaging microbiome samples?
Well, we just like to do all the major systems of the body, so like the respiratory system, the circulatory system. Like microbes are pretty much everywhere in your body, so, and it's not just a correlation between gut microbes and the immune system. It could be other microbes affecting other immune systems. So hopefully with this research we can build upon it to other systems as well.
Let's maybe take one more question. Yes.
Why did you guys choose the gut specifically?
We chose the gut because it's so, like, there's already been discoveries that link it pretty heavily to the immune system, and we thought that it was super interesting that, like a microbe, this tiny thing in one part of your body could completely affect something else in your body. So that's really why we chose this for our project.